Good morning, folks. I've got about 9:29. You might have somewhere close to that. So if you would please enter your name so I can keep the roll. Got to do that for financial aid. Let's see. Give me your name. And there we go. Riley. PJs and coffee. I hope you're comfortable. Hi, Tiffany. Shay. Chantel. Okay. Good to see you, so to speak. Fortunately, it hasn't quit on me yet. Well, I got a phone call, but I'll just have to let them leave me a message. Hi, Jessica. Hi, Tandra. Landrea, good morning. Hi, Joyce. Glad you're here. See if we can wrap up chapter, 10, I think, on the muscles? Okay. Good morning, Alexis. I may have mentioned this before, but everybody in the country now, elementary schools, middle schools, high schools, and colleges are on the internet. We just might burn it up. Hello? Yes, Asada. Yes, I am. As a matter of fact, I'm getting the names from folks that are online with me. That's correct. In your bar, your search bar up there, if you can put John Pritchett live, it usually connects everybody with it. Okay. I'm at a loss as to tell you what to do here. Do you? That sounds about right. Yeah. You could watch that if you wanted to. That's going to be pretty much the same thing we're going to go over today. Because see, I meet that class before you, so no, that's okay. Yeah, I won't count you there. Right. That's probably the best. The last one made. I'm not. I know just enough to get myself on and get myself on live, but I sure don't have an idea about why you can't get on it. Your mic could go out again, and then that might be the case. Yeah. I wish you the best then. Okay. Bye bye. Okay. Let's see. Nobby's here. And Tiffany, what is the number I can share the link? Oh, let's see here. Redial. What do I want? Directories, missed calls, received calls. Hey, excuse me, guys.
I didn't know you were going to change the phone. Never mind. I'll come back with it.
Okay, whatever you want to do.
Because that'll disconnect. So.
Okay. Tiffany, you there? Tiffany, you ready to get this number? Monica, how's it going this morning, Monica? I'm doing pretty well. It's getting kind of busy all of a sudden right here in this office. Tiffany, you ready for me to give you that number? Okay, Tiffany, it's 843 446 3506. And it's Asada. She sits right close to you. Okay? All right. Thanks, Tiffany. Appreciate your care. Exit. You know how life is. All of a sudden, 3 or 4 things come at you at one time. Doesn't happen too often, thankfully. Oh, here it goes again. Oh, no. Hey, Ramsey. Can you get on the site? Oh, good. Great. I appreciate that. Appreciate your sense of responsibility. Okay, well, in just a few minutes, we'll get back on reviewing the muscle transmission or muscle contraction, rather, and we'll see if we can wrap it up today. Okay? Thanks, Ramsey. Bye-bye. Okay. Okay, let's see. Okay, Kia, you're here. Takia. Fooled me there for a minute with just Kia. I said, I don't have a Kia. Oh, I thought I got on. That's great. Appreciate that help. Maybe that helped her out, Tiffany. Okay, Kia's here. Jalen's here. Good. How's the pain, Jalen? Got it sort of so you can handle it? Victoria. Where is Victoria? Okay. Hi, Jaquita. I was just looking at your name, wondering if you're going to make it today. Thank you. Randy. Good morning, Randy. Oh, Jalen. Sorry you got to go through that. As long as it's not infected, you're still on the high water. That's good. Okay, Rhonda. Good. Looking for Haley. Looking for Haley and Caitlin Rothwell. And Millette. Oh, Millette, thank you. Glad you're here. Boy, that's technology, isn't it? One day you have to use your phone, the other day you can use your computer. Ramsey's on television. Oh, my. Anything people put together, going to have some problems somewhere down the line. We're not perfect. That is for sure. Boy, these laptops can be persnickety, can't they? Good job, Ramsey. Keep trying. Oh, man, it takes 15 minutes just to get everybody loaded on here, doesn't it? Probably because the internet's so loaded with folks. Okay, well, we'll have to move on. Thank you, Tiffany. I'll have to remember that. Good, Rhonda. Glad it came on. Okay, let's see if we can get some information out there and wrap this chapter up. And according to what I've heard about that proctoring program that they're going to put on, it's supposed to be honor lock. And as soon as we get it, we'll put it on our system, and somehow we'll get a test out to you so you can take it. If you don't have most of you, I guess, have a computer. Might be pretty tough taking that test on your phone, but you guys are pretty good at using the phone, so I guess maybe you can pull it off. Anyway, what I want us to do is do a quick review here so we can get on into the end of it. The rest of the material that is needed to be covered. Let's go back to 343. It's going to be figure 10.4. Figure 10.4. Should see the little muscle attached to the bone up top. You see the extension of the fascicle, and then you see the muscle fiber. So look down at the muscle fiber, just getting you pictured in your mind that muscle fiber, and you see the endomysium. That's part of that dense regular connective tissue. And then you see up there at the top it says sarcoplasmic reticulum. Same thing as endoplasmic reticulum, but it's in the muscle. And then you see the transverse tubule, and then the two terminal cisternae. Come down to the little block at the right lower corner, and you see an enlarged view. Call it a triad. One T-tubule and two terminal cisternae. Remember, a cistern is a cavity. And so what you've got there on either side of the transverse tubule is an enlargement where a number of these tubes of the endoplasmic reticulum come together and make sort of a container, as it were. So you have that, and then you see, as you look right above the triad, you see myofibrils. And remember, myofibrils are organelles, just like organelles in another cell. Of course, other cells don't have myofibrils as an organelle. They have Golgi apparatus. Boy, it looks like the timing today is good on this screen, doesn't it? Yeah, much closer than it was the other day. And of course, you can see the mitochondria that are going to produce the ATP and so forth. So keeping all this picture in mind, come over to 344, the next page, where you see figure 10.5. And so you look at the figure, at the top, you see the muscle fiber again, same drawing, just minimized a little bit. And they pulled out a little section of a myofibril, just to get this in your head as we go through the process of contraction. And so you look at the enlargement below the myofibril, and you can see the about 7 proteins that are involved in that section down there. Find the Z line on the right. You see the Z line, sometimes they call it the Z disc. That is a type of protein that anchors the actin. And then you see the Z line on the other side, Could be called the Z disc. That's the protein, same protein. And you see the actin, that skinny little blue filament, that is the actin. So you've got a protein there in the Z line, you got a protein in the actin. You see the big red stuff in there. It looks like it's got little balls or bulbs on the ends. That's your myosin. There's another protein. You see the M line, which is 3 blue lines in the very middle. Okay, if you haven't found that, Ramsey says it might be on page 342. Got a different edition of the book. You might have it just a page or two off. So the M line is another protein. And as you go back to the Z line, you see the little coiled structure. That coiled structure, they haven't labeled it there in the diagram, but they call it an elastic filament. But you look below and you see an enlargement of that coiled structure, and they call it titin. It's like a spring. Now remember, a spring can stretch, but a spring can also be compressed. You want to keep that in mind because that's involved in relaxation. So we'll get to that in just a few minutes. And then if you look at the actin filament, lower right of that illustration of figure 10.5, you see the grapes, and those little balls are the actin protein. They are shaped differently because they have different amino acids, different numbers of amino acids, different sequences of amino acids, and that all helps to determine the shape of the protein. Remember, we've been saying how important it is to keep the shape of the protein. If you change the shape of the protein's not going to do what it was intended to do. So as you look at that, you see you've got the little actin. You see the little hole down below in that actin unit, subunit. That's the active site. We're going to get into that in just a second. You see the blue line right above, got a couple of spiraling blue lines of another protein called tropomyosin. Tropomyosin covers the active site, so there's no contraction. In other words, it's relaxed. The muscle is relaxed. And then you see the little yellow balls that are arranged into threes. That's another protein called troponin. Kind of makes me think of a Mickey Mouse picture. You know, we used to watch Mickey Mouse when we were little kids, Mickey and Minnie. So they had their little heads and then they had their little ears out. So that reminds me of Mickey Mouse. But anyway, troponin is another protein. Let's go now over to page 348. We're looking at figure 10.9. 10.9. You're looking at part A. Look up at the top where it says relaxed sarcomere. And you can see the M line, you can see the myosin, you can see the actin. They're just slightly overlapping. You can see the Z line. Some of the other things they don't show. Remember your bands. The I band is where the Z line is, and only the actin filament's very light. They call it the I band, isotropic. So you've got plenty of light in there. Where you get the overlap in the middle, that's the anisotropic band. So they call it the A band. So look at the lower picture, the lower half of that picture where contraction has taken place. And you see the actin filaments are almost into, or you can say they're into the M line. And you can see it sort of funnels that zone, the H zone down. It's narrow. So you can see the actin is pulled into the middle. The myosin really doesn't move, but the actin slides toward the middle. If my knuckles were the M line, then the actin moves toward that M line. So relaxed, contracted, relaxed, contracted. So that if it's contracted, you have shortened the sarcomere. I think I told you that a muscle fiber can have somewhere around 80,000 sarcomeres. Got a lot of sarcomeres in our body, don't we? All right, so let's go to page 352. 352. This is the process of skeletal muscle contraction. We talked about down at the bottom, if you come down to 352, that may be on a different page depending on your edition. It might be 350, 348, 354, something like that. You see the term neuromuscular junction. Neuro, nerve, muscular, muscle, or what's the other term that we could call that connection? What's the other term? Somebody answer that question. How about it, Millette? Millette McKay? How about give me the name of that, another name for the neuromuscular junction? That's correct, Teresa. Anybody else remember it? You're right. That's right, Teresa. Sometimes you're trying to get it in so fast, you make a mistake, don't you? Yeah, I understand. Me too. I have to slow down. It's a figure. We're looking at figure 1012, Tiffany. Millette, you're correct. Usually you put an AL on the end of it instead of an O, so it's myoneural junction. It's actually two words. You're on the right track. Okay, Ramsey, you and the Phantom need to put a myoneural AL instead of an O. So you really have two words there. Myoneural junction. Okay. So look at the figure 1012 on this page 352 or whatever page it is. I don't know if it, Ramsey, if you go on a different page, you might type it in so somebody can see that. Okay, Landrea, are you looking at 1014? Is it that what it is in your book instead of 1012? You should see a little muscle cell with a nerve coming to it, and it has three branches that come off that nerve. It says motor neuron at the top, and then you see muscle fiber, and then you have three branches, three yellow branches. Your color may be different with a different edition. Figure 1012. That's correct. That's what's listed in my textbook. So what you see enlarged down there is one of those neurons. They actually call them telodendria. We'll get into that in the next chapter when we get into, okay, good, Landrea. When we get into the next bit of the nervous system, we'll look at that. So you see there's a motor neuron. That means it's going to carry a message from your brain to your effector, whatever that might be. In our case, today, it's a skeletal muscle. So you see the little branches, and they lock onto the surface of the muscle cell. And down below is an enlarged view. So you see the axon terminal. That's that bulbous structure. Looks kind of like a, I don't know, maybe a camel's foot or something like that. There's your axon terminal. Inside the axon terminal, So 1014 for you. That's you can see one there. This one's on 1012. Shows a similar picture, but something hasn't happened that we're going to see happen in just a second. So on 1012, you see inside the axon terminal, you see the synaptic vesicle. And what is that vesicle composed of? Not the little diamonds inside. That's acetylcholine. What is the structure of the synaptic vesicle composed of? That's correct, Mallory. Appreciate that. It's phospholipid bilayer. The same thing is true of a lysosome. The same thing is true of the ER. They're all composed of a phospholipid bilayer. Now, Rhonda, as you look at what you've given for an answer, the little green diamonds, or some of you have a better idea of what color is. You might call it aqua, I'm not sure, but those little diamonds, that's the acetylcholine contained within that phospholipid bilayer. That's correct. Can't you imagine me asking a question like that on the test? Yes, Mr. Pritchard, well, you can imagine that. So, you go down to the bottom of that axon terminal. You see synaptic cleft. Remember, what is a cleft? What is a cleft? What's a cleft? A space. That's good. That's good. Space. Remember, Moses was in the cleft of the rock when God passed by him. So that's a space, and it's got sodium ions present. There are more sodium ions present in the fluid in that cleft than there are on the other side of the sarcolemma. There are more sodium ions. That's a narrow space. That's right, very narrow space, Joyce. Synaptic cleft, filled with extracellular fluid, but it's got a lot of sodium ions in it. So much so that they don't show this, but there's a positive charge on the outside of the sarcolemma and a negative charge on the inside. They just don't show that. That's a lot of sodiums on the outside, don't have as many on the inside of the muscle cell membrane. So you look to the left, you see sarcolemma. That's the cell membrane, phospholipid bilayer. And look at these little purple guys. It says ACH receptor. That's acetylcholine receptor. Acetylcholine is a neurotransmitter. And that protein that you see there, that receptor, goes all the way through the cell membrane. It's one of those proteins that goes through the cell membrane. Looking back here in chapter 3, let's see here. When we were looking at the phospholipid bilayer, and they talk about them being integral proteins, sometimes peripheral proteins, but most of the time we call them just protein. We'll just call them or transmembrane protein. That's a good term to use, transmembrane protein. So they go all the way through so that you got some on the top, some on the bottom, or I should say some on the outside, some on the inside of the cell. Transmembrane, that's good. Monica, that's good. Okay, so it's set up. Now look over to the next page, look over at the next figure, which for me is 1013. And as you look at that axon terminal, there's a green line coming down. Now we're starting to get the events that would cause muscle contraction. Anything that disturbs these events results in a failure of muscle contraction. So here's the action potential. You see over to the right calls it number one. You could call that an action potential. You can call it a nerve impulse. So the nerve impulse has been generated from up here, just like when I'm moving my arm and my fingers and so forth. I don't really think about that. I just do it. It's amazing. You don't have to say arm rise, hand open, hand close, extend. You don't have to do that. It flows like water over a dam. So anyway, here comes the nerve impulse, and as it hits the synaptic bulb, or you can call it the axon terminal, either of those terms would do. You see it causes the synaptic vesicles. You look inside it, you see the synaptic vesicles, that's ACH in the synaptic vesicle. You see the synaptic vesicle fuse with the cell membrane of the neuron. Remember, the vesicle is made up of a phospholipid bilayer. A lot of thinking went into that. So the vesicle fuses, becomes one with the cell membrane of the neuron, and out comes the molecules of acetylcholine. Acetylcholine. They go into the cleft and they attach to the transmembrane proteins. And when the acetylcholine molecules attach to the protein, the transmembrane protein, that protein changes shape. Isn't that amazing? It changes shape, opens up so that sodium can rush in. So the movement of sodium from the extracellular fluid through the protein into the muscle cell, that movement of sodium through the transmembrane protein inside the cell, that movement is due to a concentration gradient. That's possible, Jalen. That's possible. I have to look a little bit up because I think there's probably a couple of ways that can happen without you sending a message there consciously. Okay? So we'll leave it at that at this point. Okay, so sodium rushes in. Now, that means that the inside of the muscle fiber is going to get positive, and the outside is going to become negative. It's a reversal of charges. So sodium rushes through the transmembrane protein, and on the inside, it becomes positive, becomes negative on the outside. Next page, figure 1014. Okay, let's see if I can remember what I said, Teresa. When that acetylcholine attaches to the transmembrane protein opens, actually changes shape, and that's what opens it. Sodium rushes in. Remember, there was more sodium in the cleft than there was inside the cell. So sodium rushes in because there's a concentration gradient. More sodium outside than there is. Sodium moves down its gradient into the cytosol, into the cytoplasm of the muscle. So now it becomes positive on the inside of the sarcolemma and negative on the outside. Does that help you, Teresa? All right, we're on the next page. We're on 1014. Yeah, what you want to say there, Jalen, is the transmembrane protein is the receptor for the acetylcholine. Make sense? Good, good. Now we're looking at 1014, at least in my book. And it's similar to what you've been looking at. So all that we've been talking about has already occurred. Now look at the axon terminal. That's the yellow thing. Come down into the sarcolemma. You see the sodium coming in. Now notice to the right, along that green, kind of luminescent green membrane, you see the positive charges? Remember what I told you? It becomes positive on the inside of the cell and is negative on the outside. This is an action potential. It's a change in potential. And that'll be, instead of being positive on the outside, negative on the inside, it is changed. That's like a message to the muscle. So follow, going to the right, you see the positive charges as sodium is coming in, and you find that charge, that action potential, that impulse gets to a transverse tubule. That's where that big area comes in that you see is kind of light brown and got a green arrow going down in the middle of it. So you can say the impulse, you can say the action potential moves down the T tubule into the T tubule. And you see the little arrows going off. It's moving out, causing a change. We're not going to get into exactly how that change occurs, but I want you to look at the two blue structures on either side of the T tubule. What are blue structures? Look at the diagram. See if you can determine that. What are those two blue structures? So many shades of blue. I don't know what shade you'd call that. That's right, Teresa. Terminal cisternae. Got two of them. And there's the tubule in the middle. So therefore you have the triad. Now look at the right hand one. Look at the right-sided terminal cisternae, and you come halfway down, you see calcium +2 channel open. This is what the action potential does. It opens calcium channels of the terminal cisternae. So the impulse, the action potential. That's right, Jerkita. You're right. That's correct. That change, you could call it that too, Tiffany. smooth endoplasmic reticulum, okay, and the terminal cisternae. See, calcium is inside those. Calcium is inside the terminal cisternae. And when the channels open, calcium pours out into the cytoplasm of the myofibril. That's right, Alexis. So you got calcium in both of the terminal cisternae. The impulse comes down the T-tubule. The action potential comes down the T-tubule, causes the calcium channels to open, and calcium comes out into the cytoplasm. If you want to put cytosol, takes fewer letters to say it, and that's okay. Calcium, okay, Jalen, calcium's present in the terminal cisternae. In a relaxed muscle, it's sitting inside the terminal cisternae. But when that impulse comes down the transverse tubule, you see those little pluses coming down, they open the calcium channels of the terminal cisternae. You can see they got little transmembrane proteins down there too. Man, do we need proteins. That's why you got to eat some good protein. Eat some cheese, some chicken, things like that, chili. That whole structure, along with the tubes going that you see sticking out like open pipes tie, that whole structure is called the Sarcoplasmic Reticulum. It's basically the same thing as the endoplasmic reticulum, but it's modified a little bit in terms of what it contains. There's nothing wrong with calling it the Sarcoplasmic Reticulum, but these two sections on either side of the T-tubule, they have chosen to name them terminal cisternae. That's about all I can tell you. I have to read up on how they decided they wanted to name that. I hope that helps you a little bit. But both of them would be called Sarcoplasmic Reticulum. You wouldn't be wrong to call it that. But that particular section of the Sarcoplasmic Reticulum tie is called the terminal cisternae. Just an enlargement, like a big vat full of calcium. That's why you got to have milk, got to eat yogurt, got to eat cheese, and all those good things that got calcium in, got your minerals and so forth. Okay, so we've got the calcium now out into the cytoplasm. Now look over on the next page, figure 1015. I tell you, this is just fascinating to me. Man, this is complicated, isn't it? Yeah, it is flat out complicated. I don't know what BG plus means, Andrea. You'd have to tell me what BG plus means. I'm at a loss for that, kiddo. Okay, so we got calcium out into the cytosol, right? Look down at the bottom of the next page where you see figure 1015. Look at the bottom of the figure, little rectangle or square maybe. And what do you recognize in there? Well, you see a myosin head, and then you see those purple grapes. And if you look very carefully, you can see that tropomyosin covers those little holes. Those little holes, those little indentions, those little depressions are the active site of the actin molecules. Jerkita, you're halfway there. You got to have a third structure. You got two terminal cisternae, and you have one transverse tubule. So that's three things. Like my middle finger would be the transverse tubule, and these two fingers would be the two terminal cisternae. Okay, Lindrea, thank you. I know they got all kinds of language, so I just haven't been exposed to that. Thank you, Lindrea. So the calcium's in the cytosol, or the cytoplasm. Look at that little square at the bottom of the page, or bottom of the figure 1015. You see the tropomyosin, that's the light blue protein, different shape, different amino acids, different sequence of amino acids. You see it covers that little dark area, which is a depression. It's the active site of the actin. Now notice you got three little yellow balls there. Troponin, like we said, those are globular kind of proteins. Different amino acids in a different sequence, different numbers, get a different protein that carries out a particular responsibility. And sometimes it's fun to think about this, although my wife would probably not appreciate it very much. When you and I cook a steak, or have hamburger, or have sloppy joes, or chili with some ground up beef in it, that's what we're eating. We're eating the troponin, we're eating the tropomyosin, we're eating the actin, we're eating the myosin. All we do is cook it so it breaks it up a little bit, easier for us to chew. And then we digest it in our stomach, in our small intestine, and we turn it into our muscles. Gosh. Oh, and we don't have to think a thing about it. Our body runs on autopilot. So we can talk to each other and just have a good time being together. That's correct. That's correct, Jerkita. That's correct. Now, Jerkita, are you looking at the bottom box on figure 1015, where you see the actin and the tropomyosin and the troponin and the myosin head? You're looking at that? Or are you still back over there on the previous diagram? I think purple might be mixing us up. Jerkita, go back to page 343, figure 10.4. And those of you that are interested in it, go back so you have it, get it straight. Okay, are you back on 343, figure 10.4? So as you look at figure 10.4, notice what they put the pointer on that big muscle fiber there. Oh, you're caught up. You're okay? All right. So we're looking on at 10.15, and you see the little actin balls. That's protein. You see the tropomyosin, which is covering the active site. You see the troponin, which is in position. You see the myosin head. Now look to the right. Remember just what happened? The calcium channels of the terminal cisternae released calcium. Look at the little Mickey Mouse head and ears in figure B. You see the little balls there? There's a couple of calcium molecules. Calcium binds to troponin. You don't have any calcium, you're not going to have any contraction. So the calcium combines with the troponin, and look at the blue tropomyosin. You see how it's moved so that the active sites, that's the little depression, is exposed. This sure sounds cumbersome, doesn't it? But it happens in a flash. That's why somebody like Usain Bolt can fly down the field. You're correct, Jerkita. You're correct. You're correct. Okay, everybody's looking at that square now. So you got a couple of calcium molecules on the troponin. It causes the tropomyosin to move. Now the myosin head can attach to the active site. Good, Jerkita. Now I want you to look at figure 10.16. It shows a relaxed sarcomere at the top of the page, figure 10.16. You can see there's a bit of an overlap in there from the actin overlapping a little bit of the myosin. But look at the next line down, next box, and you see the myosin head has attached to number one of those actin proteins. Number two, you see it says power stroke. When you go down to the next block, you see that myosin head is pulling on both sides. You got myosin head on the left, myosin head on the right. It's pulling the actin into the M line. You see the word M line up there at the top, and you come down with that arrow right in the middle. Pulls it in. Another myosin head is attached to the second actin protein, pulls that in, and so you can see it's moving the actin in toward the M line. That's contraction, shortening of the sarcomere. So when you look at the bottom, you see what a contracted sarcomere looks like. Everybody okay in terms of contraction? So now we want to relax, don't we? I want you to look on page 358. And you should see a heading that says muscle relaxation. So we've gotten that sarcomere to shorten. The Z lines come closer together. Does everybody see the muscle relaxation? We moved over, I think, to another page. I'm not sure, you might have a little different arrangement of things, but look for a section that says muscle relaxation. There's a couple of things I want you to know there. Number one, as you look under that heading of muscle relaxation, you see a big long word. Now, it's actually got acetylcholine in the word, but you see STERASE. ASE on the end of a word means what? Mm, 358 for me, Chantel. Let's see. Jerkita, when you say doing the pulling, it's the myosin heads that are pulling the actin. The myosin heads are contracting. I'm not looking at figure 1018, but Ty, that's right. You see ASE on the end of a word, acetylcholinesterase. That's a mouthful, isn't it? That's an enzyme that's going to break down acetylcholine. That's correct, Tiffany. That's correct, Alexis. So if you break down the acetylcholine, what are the transmembrane proteins going to do? They're going to close. No more sodium rushes in, no more action potential. Come down to step number 3. You see it says calcium ions are pumped back into the SR, or you could say back into the terminal cisternae. And you come down to number 4. It says troponin and tropomyosin block the active sites. Now the muscle is relaxed, or the sarcomere is relaxed. So now you're relaxed. At least that muscle is, so the sarcomere is relaxed. So Riley, when you want to get up and get a cup of coffee, all that event to make your muscles contract, your back muscles, your thigh muscles, and so forth, your hand and arm muscles to put the coffee in and the water in and so forth, that's happening. And as you quit doing that, they relax because acetylcholinesterase destroys acetylcholine, no more action potential. Calcium is pumped out of the cytosol back into the terminal cisternae, and troponin and tropomyosin block the active sites. And so you've got your cup of coffee and you're sitting back in the chair and there's some relaxation. And some of the muscles you just used to go out of the chair, go to the kitchen, make your coffee, come back, and now you're sitting down. It seems so cumbersome, doesn't it? All that activity. And yet it happens in a flash. so quickly that it's very coordinated, whether you're playing a piano piece or whether you're walking to the kitchen to get a cup of coffee. Graceful contraction. Yeah, that's right. That's something that's an integral part of your life, isn't it? Getting some good cup of coffee. Mm, 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 mm. Let's go over to page 361, and we're looking at a section on the second column. It says immediate sources of energy for muscle contraction. Immediate. I want you to read that paragraph. It's about adding a phosphate group to adenosine diphosphate. You know about adenosine triphosphate. So just go down as far as to where you go right under that little formula where it says ATP plus creatine phosphate. The CK is an enzyme, and then it produces ATP and releases creatine. That's where we get some energy. Now we're going to skip over to section 10.7. It's a section, like a category within the chapter. I'm on page 368. Actually, I'm going to say this. It happens also in your heart, but you've got a little system in your heart that sends out a message to make the heart rhythmically contract, 60, 80 times a minute. So we're applying this to voluntary contraction, but you have a similar situation going on in your heart, but you're just not thinking about it. So get to 10.7, and 10.7 is a section of the chapter, and it's labeled muscle tension at the organ level. Muscle tension at the organ level. Now, under that heading, muscle tension at the organ level, look down in that column, and you see motor units. Motor units. Everybody seen that word, motor units? That's the heading over a little section we're going to talk about for just a few minutes. Well, it would basically be the same function in the heart. The heart's got a few anatomical differences, but you've got a center in your heart, a little control center that sends out an action impulse or action potential. And so it makes all the muscle of the heart contract. Just like the same kind of mechanism here, but you don't have to think about it. Aren't you glad? Some of us would get so mesmerized in a movie or listening to some music, we'd die, wouldn't we? That's it, Jalen. Yep. You'll get that in the next semester when you get into the cardiovascular system. The essay no. Everybody found muscle tension at the organ level? And you have motor units. Come into that paragraph. The second line down says a single motor neuron, along with its muscle fibers, leaves out a word like which it innervates, is called a motor unit. You want to know that definition. Motor unit. It's a single motor neuron along with the muscle fibers which it innervates. In other words, it connects with those muscle fibers. Now you can see an illustration of that as you look on page 369, figure 1025, and the name of that figure is the motor unit. Now, as you look at the illustration, you see over to the top right, you see it says spinal cord. Looks like it's got something like a butterfly or a moth in the middle of that cord, doesn't it? So that's cross section of your spinal cord, and you look at the red and the blue coming out of that little section of that darker brown. It's sort of surrounded by the lighter brown, the creamy kind of beige maybe. And you see you've got a couple of neurons going there. Those again are motor neurons. We haven't gotten into axons and dendrites and all that, but you can just call it a motor neuron. Now follow the blue one. Follow the blue. That's true. Don't worry about that right now, though, Ramsey. So the blue, as you see it curves around, it starts to branch. Yeah, that's no problem, Riley. You might have to call me up or something like that. But anyway, notice the branches, and you see the branches go to, in that case, blue muscle fibers. You see the red comes out, and it branches to, and it innervates some other muscle fibers. It's got a lot of connections between your nervous system and your muscle fibers. Now, go back to that paragraph where you saw motor units. And you just highlighted, wrote down something about a motor unit, definition for it. Come to the next paragraph. An average motor unit consists of about 150 muscle fibers. That means one neuron innervates 150 muscles. Now notice the rest of the sentence, but this number can vary widely with the degree of motor control needed for the muscle. Keep reading. Muscles that require fine control, like our fingers in painting or writing or tying a bow for your shoes and then doing a square knot over it so it doesn't come undone and trip you up. Muscles that require fine control, like your hand or your lips and your tongue. And it mentions here also such as those around the larynx, your voice box, and it mentions fingers, will have multiple small motor units, often containing as few as 10 muscle fibers per unit. One neuron works 10 muscle fibers. So that's why we can do very intricate work with our fingers. And think of all the sounds that we can make in a language with our lips and our tongues. We don't think about it. We just practiced it when we were kids until we got it right and said the word correctly. So your lips, your tongue, your fingers, and some people even seem to develop it in their toes. Those who have lost their hands. Now, come on down in that same paragraph. You see it says as few as 10 muscle fibers per motor unit. This gives the nervous system more precise control over the amount and rate of tension produced. Conversely, large, powerful muscles, such as your postural muscles, your gastrocnemius, your thighs, and so forth, big muscles like that can have 2,000 to 3,000 muscle fibers in one motor unit. That one neuron branches to 2,000 muscle fibers because you want them to all contract at the same time. Those are big muscles used for walking and jumping and swimming and climbing. These guys are for fine movement, playing a piano, playing a guitar, mandolin, or something like that. And when you have to pick up something, say with your biceps, if it's real light, like this pen, you might activate only 50 muscle fibers. But you go put a brick in your hand, and it's going to weigh many, many times more than this pen is. It might take, instead of 50, it might take 350 muscle fibers to pick that up. Now, we've learned over time that a brick weighs more than the pen. So in our minds, when we pick it up, we only activate the muscles needed to pick that object up. So we can use a few of these muscle fibers because we know how much that pen weighs versus the brick. Well, we're not going to get through this chapter yet. But the last thing I want you to do is look on 369 where you see the heading called muscle tone. I want you to read that sentence, 2 sentences right there with the word muscle tone. Even when a muscle is at rest, it still has some degree of tension, and that's called muscle tone. Doesn't move anything. It's just got a little bit of tone to it, a little bit of a contraction to it. Come down to the next paragraph where you see the word hypotonia, which means that the muscle is not as toned as it should be. It's more flaccid and soft. Sometimes neurological damage will cause a muscle to just be completely relaxed. Sometimes we have a problem where we have too much tone, hypertonia. Well, we got just a little bit more to cover, and we'll do that on Tuesday morning at 9:30, Lord willing. Everybody okay? Oh my, let's see. Where were we? Are all of you there with Riley's remark? Where were we when the system froze up? Maybe I covered it when I did that little review on the contraction of the muscle. That's true. Can you tell me where it froze in terms of looking at what we covered, the material covered? Okay, Andrea says 1016. So, okay, we went over that particular figure. So, do you feel comfortable with what the myosin is doing to the actin? Maybe we got up to that point where the calcium hooked onto the troponin, and the troponin caused the tropomyosin to move, so active sites on actin could be attached to by myosin. All we're doing there is showing how the myosin pulls the actin into the M line, which is shortening the sarcomere, or we would say contracting the muscle. Okay, so do you feel comfortable now about what we talked about this morning on figure 1016? Did I elaborate on it enough to where you understand the movement of the actin toward the M line? Joyce, you said we covered it today. Okay, Jalen, you think you're somewhat comfortable about the action potential and so forth? Covered it at the beginning of this session. You feel comfortable with that, Jalen? Riley, you're satisfied? Feel you got it together? How are you going to get all this in your head? Somebody's going to ask you some questions about it. Teach your closet door. Know the sequence. Okay. All right, I'm going to close this out, guys. So do your social distancing and things like that. Keep your hands away from your mouth and your face and your nose, and keep healthy. And Lord willing, we'll see you come Tuesday at 9:30. That's what you got to do, Jaquita. You got to go back and listen to it and think about it. So many steps could go wrong. But most of us, we can move where we want to move, can't we? Fascinating stuff. That's good. Okay, Ramsey. I'm going to close out, guys. See y'all later. Bye-bye.